Autoimmunity definition: it's an immune response against the self-antigens and is important cause of autoimmunity is an immune response against self-antigen and is important cause of death. 5 to 7% of adults in Europe, North America, two-thirds of are females. More than 40 human diseases are of autoimmune origin, such as SLE, RA, and IDDM. A small number of B cells which reactive to self-antigen are always present in the lymphoid organs. If regulation of these autoreactive cells break down clones of lymphocyte can generate high level of autoantibodies or autoreactive cell leading to autoimmune damage. Spectrum of autoimmune response. Autoimmune potential present in every body reflect T and B receptor diversity, healthy response. Physiological autoimmunity sporadic related to transient tissue damage may have a homeostatic rule. Pathological autoimmunity rare result of complex interaction of genetic and environmental factor. Categories: organ specific, the autoimmune reaction are directed against one or more specific cytoplasmic constituent, plasma membrane structure, or secreted product of the cell. Non-organ specific, the autoimmune reaction are directed against a structure common to many tissue and found through the body, like nuclear component, mitochondrial proteins, or constituent of muscle. Here we found a table. Myasthenia, the spectrum of autoimmune disease, the disease and the principle of autoimmunity. Normal or physiological autoimmunity. More than one abnormality are required to induce autoimmune disease. A heterogeneity of tissue, destructive mechanism, diversity of initiation agents, the multifactorial pathogenesis of the autoimmunity. Normal or physiological autoimmunity. It's not true that all autoimmune responses are bad cause diseases. Some autoimmune responses have important physiological function, are essential for the normal functioning of the body. Example of normal autoimmunity: the action between CD4 and CD8 T cells and the measure histocompatibility class 2 and or class 1 respectively. Anti-idiotype antibodies: the antibody binding site idiotype on immunoglobulin molecule can function as epitope, provoke production of antibodies. Anti-idiotype: each anti-idiotype response tends to exert a negative feedback on the preceding response. Therefore, the net effect is termination of antibody response. Cell-cell interaction mediated by adhesion molecule and their ligands. Removal of the aged RBCs as RBCs become aged. An onion transport glycoprotein called band 3 will be cleaved. A new epitope will be formed, which will be recognized as autoantigen. Interaction between these antigen antibodies will facilitate phagocytosis of RBCs. The band 3 protein is also present on platelet lymphocyte polymorph and relay. More than one abnormality is required to induce autoimmune disease. It's more likely that more than one mechanism is impatient is dysregulated before an immune response acquires pathological significance. Heterogeneity of tissue destructive mechanism. Multiple different cells types such as anti-specific lymphocyte, non-specific phagocyte, and granulocyte fibroblast infiltration autoimmune inflammatory lesion. Several agents can initiate an immune response in the microenvironment of the affected tissue. The genetic background of the patient finally lead to pathology. The multifactorial pathogenesis of autoimmunity. Autoimmune diseases develop in hosts with multiple inherited risk factors combined with environmental contribution. Factor affecting severity of autoimmunity. Quantity factor, how much autoantibodies present, how great the T cell response, how much of the cytokine produced. Quality factor, antibody affinity, isotype, fine specificity. Genetic factor, either augmenting or allowing susceptibility. Environmental factor, infection, chemical, food, induction of autoimmunity. Many autoimmune diseases show spontaneous remission exacerbation, suggesting an unstable relationship between positive and negative and suppressive cytokines. Initial triggering, formation of autoreactive cells, T cells, normal pathway, special circumstances, gene, complement, hormone, negative selection, no problem, failure to negative selection, autoreactive T cell or autoantibody, tissue damage, and autoimmunity, and autoimmune disease, and autoimmune patient. The generative lymphoid organ, abnormal selection or response, self-reactive lymphocyte, cytokine, other stimulus, presentation of self-antigen to autoreactive lymphocyte, tissue injury, and autoimmune disease. Theories of autoimmunity, failure of thymic education, breakdown of peripheral tolerance, antigen non-specific lymphocyte activation, molecular mimicry, abnormalities in lymphocyte interaction, hormonal factors, left handedness, environmental factor, exposure to of hidden and failure of thymic education, removal of self-reactive lymphocyte from T cell repertoire is achieved during thymic selection through colonal cell deletion or energy. Escape of autoreactive T cell from the thymus could supply harmful T cell to peripheral lymphoid tissue, rheumatoid arthritis, breakdown of peripheral tolerance. Peripheral tolerance is considered as a security system to prevent activation of escaped self-reactive T cells. Failure of peripheral tolerance may be due to apparent expression of co-stimulatory in tissue, antigen presenting or mutation of CTLA4 may result in breakdown of T cell energy and the tissue-specific autoimmune reaction. Mutations that interfere with the apoptotic death of mature lymphocyte may result in autoimmune diseases such as mutation in Fas and Fas ligand. 3. Defect in T cell mediated suppression may contribute to self-reactive lymphocyte inflammation, innate immunity, inflammation. Here, influx of self-reactive lymphocyte into tissue, activation of autoreactive lymphocyte, tissue injury, autoimmune disease. Environmental factor. Environmental factor virus as inducer of autoimmunity. It has been suggested that virus, especially those infecting lymphoid tissue, may interfere with immunological control mechanisms. The ones of IDDM in children, Coxsackie virus, the presence of Epstein-Barr virus, EB virus genome in salivary gland, or Sjogren's syndrome patient, presence of type C retrovirus or paramyxovirus in SLE, genetic susceptibility for autoimmunity. Family and genetic studies indicate that many genes affect the development of autoimmunity. Evidence the relative of patients with Hashimoto's disease have anti-thyroid antibodies may develop thyrotoxicosis or other autoimmune diseases. Studies with identical twins show that when one has multiple sclerosis, the other has a 30% chance of developing the disease. Genetic risk factor, autoimmune disease, immune response gene, polymorphic HLA sequence, complement gene, T cell receptor, immunoglobulin gene, cytokines genes, and gene controlling antigen processing presentation, non-immune genes, mutation in any of the gene regulating programmed cell death, gene regulating response to tissue injury, gene regulating tissue repair, has defect in cytokines production or signaling that can lead to autoimmunity, the cytokine or the problem, the defect and the result. The factors that influence the pathogenesis of T-cell mediated autoimmune diseases, genetic susceptibility, activation of the autoreactive T-cell antigen-specific antigen, non-specific infiltration of the target cells, damage to the target of HLA disease association. It's now firmly established that HLA gene influence susceptibility for large group of diseases such as RA, HLA-DR14, MS, and HLA-DR2, SLE. Hypothesis of HLA disease association. Hypothesis 1. Peptide selection model suggests that HLA allele are essential molecule for binding of initiation antigenic peptides, which can induce a virus T-cell response combined with tissue injury or would fail to stimulate T-cell response. Hypothesis 2. Molecular mimicry model suggests a sharing between HLA structure and the infectious agent. Hypothesis 3 suggests that TCR are responsible for autoimmune. The HLA allele associated with disease is due to its association with TCR. Gene is linking disequilibria rather than HLA gene are disease relevant such as cytokine and transporter genes. Mechanism of immune mediated tissue destruction. Type 1 immediate hypersensitivity, IgE bind to the mast cell and when exposed to antigen they promoted released mediator facilitating the clinical manifestation of the allergy. Type 2 antibody mediated disease, autoantibodies with complement or cytotoxic T cell can cause lysis of the cell such as autoimmune hemolytic anemia, autoimmune thyroiditis, immune thrombocytopenia, myasthenia gravis. Complex is formed by specific antibodies and the antigen combined deposited into tissue and cause an intense inflammatory reaction such as SLE and vasculitis. Type 4 T cell mediated. Many lesions in the autoimmune disease are heavily infiltrated with mononuclear cells. Autosensitized T lymphocytes contribute to pathogenesis of disease such as IDDM, ulcerative colitis, MSE, therapeutic implication. Therapies are needed that not only prevent T cell reactivity but also establish tolerance once the autoreactive T cells have been activated and an autoimmune disease is diagnosed. One example of immunomodulation is the treatment of patients with hemophilia. In about 30% of the patients with hemophilia A who receive infusion of factor AA8 concentration, systemic injection of deaggregated antigen induce tolerance in helper T cells and can prevent experimentally induced autoimmune encephalitis. A peptide that inhibits the activation of T cell clone is called an antagonist. Some viruses escape the host immune response by producing such antagonistic variant of important antigen, and the administration of antagonist peptide has prevented autoimmune encephalitis in mice. Immune modulation, because treatment with antibody against molecule important for the T cell function, induced the T cells to become unresponsive in rodent. Several of these antibodies have been evaluated in clinical studies. A form of CD152 inhibit the interaction of the co-stimulatory molecules CD8, CD86 on antigen-presenting cell with their shared receptor. CD28 on T cell treatment with this form of CD152 prevent or ameliorated lupus, autoimmune encephalitis, and arthritis in mice. T cell can be categorized according to the cytokine they produce. Type 1 helper cells produce mainly interferon, tumor necrosis factor, and the interleukin 2, where type 2 helper T2 cell produce mainly interleukin 4, interleukin 5, and interleukin 13. Since multiple sclerosis and type 1 diabetes are mainly mediated by T1 helper cells and allergic disease by T2 helper cell, alternation of the cytokines balance is an appealing therapeutic possibility. Clinical trials that are evaluating the immunoregulatory cytokine interleukin 10 with chronic inflammatory disease have reported encouraging initial results in patients with psoriasis. Antagonizing the inflammatory cytokine tumor necrosis factor with monoclonal antibody or soluble decoy receptor have proved remarkable effective in treatment of rheumatoid arthritis and Crohn's disease. Purging autoreactive T cell from the T cell repertoire. Purging autoreactive cells from the autoimmune repertoire is an option in several cases of autoimmunity. Autologous stem cell transplantation has been tried in patients with rheumatoid arthritis, psoriasis, and systemic lupus erythematosus with variable success. It's not yet possible to judge the long-term efficacy and safety of this approach, and it's unclear where the patient who receive such treatment can generate completely new repertoire of the T cells. Nevertheless, purified CD34 hematopoietic progenitor cells that are depleted of autoreactive lymphocyte and inflammatory leukocyte may remind to reduce tolerance